Okay, so we're recording, and welcome again everyone to this workshop, this Queen Bee workshop. We are Hives for Humanity Society. We are a nonprofit that was founded in 2012 in the downtown east side of Vancouver. And we work in partnership with many different organizations and businesses, community centers, nonprofits, and also restaurants and retailers who feature our honey. And we do that to create meaningful opportunities for people to connect to nature and to connect to community and to connect to really opportunities to share their voice and share their experience and be a part of communities of care. And we center those around the bees and the gardens that surround them. And yeah, my name is Sarah. I'm a co-founder and currently the executive director of Hives for Humanity Society. I've been a beekeeper since Hives for Humanity started, which was June 2012. And I've been really lucky to learn along the way from all kinds of beekeepers, including my mom, including Mark Winston and Heather Higo and Paul Van Westendorp and local beekeepers who've contributed their knowledge to our work. And also more recently to Phil Laflamme and Leslie Williams, who are our beekeepers on the call tonight. And so I'm really going to be more in a moderator role tonight and passing questions over to them and to their expertise. So I wanted to start by introducing them a little bit. So Leslie is on the right here, and this is at our Italian Cultural Center apiary, where we were doing much of the work that is featured in the photos and the videos that we'll be sharing. Leslie is the Hives for Humanity apiary manager, and she's been in that role for 2 seasons with us now. She's also the assistant director and lead the Sharing Farm Society in Richmond. And she's also on the board of the newly founded BC Native Bee Society and has been beekeeping for 9 years. So thanks, Leslie, for being here and for sharing your experience of the bees and of queens. Along with Phil. Phil Laflamme is the Hives for Humanity mentorship beekeeper, which means that we all get to learn from him and his depth of expertise, which extends back to 1975, which is super cool, which is over 40 years of beekeeping. And Phil has worked as a research technician, he's worked as a bee inspector, and he's also had 3 of his own beekeeping businesses in different provinces in Canada. So a welcome and a thank you to Leslie and Phil. And again to Diane, whose contact I'll share at the end, because there is a beekeeping club at the Dunbar Community Centre if anybody is in that neighborhood and wants to get involved. If we were gathering in person, which we love to do, and we hope to start returning to in ways that are safe and inclusive and that celebrate the diversity of bees and people in our communities, we would always be starting by gathering in a circle. And so, although we are not in a physical circle right now, I do find that in the bees and also in the ways that we gather online, there are these circles that happen, even in the way that, I see some of you kind of like leaning in to your computers and in the way our conversations move and in the way the bees move. So just taking some time at the beginning to think about being in circle together and what that means. And that usually I would be gathering us around the smoker and starting to think about what it means to be on this land together and doing this work of beekeeping and pollinator gardening together. So taking time now at this point of introduction and welcome to think about this land that we're on that is unceded and that is the land of the Musqueam and the Squamish and the Tsleil-Waututh nations, and to really be thinking about what that means to each of us and how each of us are working to be respectful of the land and of those peoples, and to build relationships with the land that are reciprocal. And as beekeepers, really always wanting to start from a place of thinking about what it means to be working with a species of bee who are not native to North America even. So to be working with one of 20,000 species of bees that exist in the world. And tonight, even getting like more zoomed in to one of the bees that we find in a colony of 60,000 bees. So yeah, I'll just pause and take a breath as I think about what that means to me. And always I'm welcoming of conversation about what that means to you as well. So Yeah, here we are in this circle, gathering around a smoker. And often at Hives, what we do as we prepare our smoker, really breathing and calming and grounding into this practice of lighting the smoker, gathering our tools, gathering our presence, and moving into really a conversation with the bees as we enter the apiary and as we enter the hive. And in our yards, we grow all kinds of medicines for people. So there's sage and peppermint in this smoker, knowing that those medicines of the plants feed the bees, they feed our community, and they feed the land as well. And as we close that smoker and as we get into our veils and as we move into the apiary together, really we are immediately starting to think about how we're moving and how we're connecting, and how we are in relationship with these bees, these honeybees. And so, I'm just going to jump right into it, and we're going to start really delving into this world of queen bees. And you are welcome to be here at whatever level of beekeeping you are. We did call it an intermediate class, so we will be getting into some of the really exciting and very complex details of what it means to be a queen bee and how we as beekeepers understand and work with queen bees. But we will start a bit gently with some more gentle introduction level. Who is a queen? What is a queen? What is her life? And so, yeah, I'm gonna pass it over to Phil and ask him to share what is a queen, Phil? What's her life cycle? What's she up to? And how is she in relationship with the drones and the workers and the rest of the hive?
Okay, well, what differentiates the queen from the workers and the drones is that she can lay eggs. She can lay fertile eggs, and she lives a lot longer. Now, the longest living queen I've ever seen is 5 years, and I know that because I marked her originally and kept track of her. I've heard accounts of queens living 8 years, but that would be incredible. Her development period is shorter than workers. It's condensed into probably 16 days as compared to the normal worker that would be 21. So she has a shorter larval period, a shorter pupal period, and she will emerge approximately 16 days after the egg that becomes her was laid. And after she emerges, she'll be in the hive. The workers would not pay much attention to her for the first couple of days, and she's in a process of going through her sexual maturity, and she'll do that for about 5 or 6 days, at which time, if the weather's appropriate, and that would be if it's over 20 degrees Celsius and the winds are less than about 20 kilometers an hour, she will take a couple of orientation flights, and then she will fly to a drone congregation area and mate with drones. And if the weather's really good, a day like today, you could classify as a good mating day for queen bees. What you're looking for is a sunny day without much wind, and the temperature being very steady. And she can get all her mating done in one afternoon. She can mate with as many as 15 to 20 drones. She can go out 2 or 3 times to do that. And the feeling is that she just mates with as many drones as she needs to until her spermatheca is full. Then she comes back to the colony, and she will not lay right away. She goes through about maybe 2, 3, 4 days before she starts to produce eggs and begins to lay. And sometimes that's a difficult period because you're not sure if she's mated, you're not sure she's in there. But what it just requires is patience, and eventually you'll start to see eggs. And that's approximately 2 weeks after we introduce mature queen cells that are about 14 days old from egg to the cell. And the cell we put in will hatch in about 1 or 2 days. And so we sort of time it. A week later, if the weather's good, she should be mating. And a week after that, we can go in and take out a mated queen. That's if the conditions are ideal.
Yeah, and I think the timing, like from everything I hear from you and Leslie scheduling and from all of these pieces that you've just said, is like the timing is so important. So often like being patient, I think, is also so important, like waiting those 2 weeks for your queen. I want to show a video, and maybe I'll just in case, like nobody has asked which one the queen is, so that's a great sign for this intermediate course. But here she is with the yellow dot, of course. So this is a beautiful queen from our Hastings Folk Garden site on the 100 block of East Hastings, where we have a community garden, and drones as well on this frame, and workers too. So I'm going to share this video, which was taken recently, which is showing some of that, showing the queen and showing her eggs, showing her laying. So Phil and Leslie, maybe Leslie, do you want to talk about what we're seeing here? Oh, sorry, I'll move it back to the beginning. There we go.
Sure. Well, you can see in the back of the cells, you can already see the eggs that she's laid, the little white speck at the back there. And probably around the edges, yeah, you can see a little bit more. You can see royal jelly and a little bit of larva in the other cells. And then, Sarah, can you get rid of the bar on the base there? Oh, there you go. And then you can see the queen is putting her abdomen into the cell up top, sort of in the top right third of the picture there. And she's laying an egg as her retinue of workers surround her. And then she's moving on, and it's hard to see in here, but she would be at different points checking with her forelegs the depth of the cell to make sure which kind of egg to lay. So these are all worker cells. And she's going around, checking again.
Yeah, and so hopefully everyone caught her there, and I can play it a bit more if anyone didn't, but this queen is an unmarked queen, so a bit harder to spot. Hasn't been marked yet, with her long glistening abdomen. And then Leslie was mentioning, I will go back to the beginning so we can see some of that glisten even in the still here. The single egg in every cell, upright, tiny, and in the center. And then in some of these cells over here, those eggs having hatched and now swimming in royal jelly. Phil, could you talk a little bit about royal jelly and what it is and who gets fed it when? One of the things that I've never really been sure of is, when do workers stop getting fed royal jelly, and what is worker jelly?
Leslie, I think you were going to talk about that, weren't you? Didn't you have
Great.
Sure, I've got some info on royal jelly. So it's secreted by the hypopharyngeal glands of workers, usually when they're between 5 and 15 days old. That's one of their jobs, is to feed royal jelly. And proteins are the main substance of royal jelly. It also is very rich in amino acids, 8 essential amino acids. And it also has a crucial antibacterial, antifungal, antimicrobial property, which keeps it clean for the bees. And it's fed to the queen throughout her larval and adult life. And it's also fed to young workers and to drone larvae, young worker larvae and young drone larvae. So you want to add anything to that?
No, it tastes bitter. If you were to sample royal jelly, it has a bitter taste to it. And I know it's high in vitamin B12, but you're probably better getting your vitamin B12 from another source than from royal jelly.
Yes, it's quite hard to actually harvest much of it. We do collect some for queen rearing, and we do a whole round of grafts of larvae just to get the workers to fill those cups with royal jelly, which we then scoop out. It's quite painstaking. I know there are a lot of cosmetic products and health products that have royal jelly in them, and I'm sure it's a very tedious job to do, to collect a lot of it.
It definitely is one of the joys of keeping bees, though, is that, you know, if you ever do have unwanted or an extra queen cell with royal jelly, and I always scoop some honey with it because it's very bitter. But this magic too. Diane was asking if either of you know what chemical is the antifungal one. That might be a question for the Mark Winston Bee Biology appendix or index. That's often where I go for so specifics, but yeah.
I don't know it off the top of my head, unfortunately.
No, I don't either.
Yeah, I'll go to that appendix afterwards and try and learn that one. And Alyssa is asking, yeah, if when it's sold as a supplement, like sometimes you see it in little vials, would it ever be fabricated? Like, is that a thing that can be synthesized? I know that people have tried to synthesize beeswax, and just the complexity is so incredible that that hasn't been possible.
I don't know if it could be synthesized, but it could certainly be adulterated. There are some beekeeping operations, a lot of them in Asia, that's all they do is they harvest royal jelly, and they have actually machines that they can attach to the cell at the right age, and it separates the larva from the royal jelly, and it's just sucked out on a commercial basis. But as far as I know, there's no CFIA requirements on a jar of royal jelly as attesting to its purity or not.
And so can we just clarify too, so yeah, all bees get fed royal jelly for the first couple of days of their life after they hatch. And then the queen gets fed it her entire life as an adult as well. Is that true?
I've always, recently I've heard more of this mention of royal jelly fed to workers. I've always used to refer to it as brood food. I think it's sort of a thin royal jelly. It doesn't have all the ingredients that the food fed to the queen would have.
Yeah. And where most of the workers are getting fed brood food, which is being created from pollen as a protein source, and nectar as a carbohydrate. The queen bee is getting fed this excretion. Leslie was mentioning the hyperpharyngeal glands of the worker bees. That's a gland in the forehead of the bees that is primed when they are sort of 1 to 5 days old. So they secrete the royal jelly into the cells that feed the larva, and then they also feed that to the queen bee for her entire life. So when we see that retinue, that gathering of workers around the queen, those are female worker bees who are grooming, who are cleaning, who are feeding, who are, you know, taking care of that queen. And so let's move into this next slide showing some of the queen cells where we would find that royal jelly. Yeah, you know, when do these cells occur in a hive? Like when we see queen cells in our colonies, what kind of indications are they to what might be going on in the colony? Like when does a colony make a queen? And then what are the interventions that we as beekeepers can make at those times? Leslie, I think you were going to start us off with this.
Yeah, so I'm sure many of the beekeepers on the line have found swarm cells in their colonies before, which is when, of course, when the colony is very strong and they've decided to reproduce themselves through swarming. So those typically would be found on the bottom of the frames, and often there would be quite a lot of them. We recently found 30 in one colony, so we were a little bit in awe of that for briefly. We dealt with that pretty quickly. And then another kind of cell that you might find is an emergency cell, which is when something has happened to the queen abruptly. She's been killed, perhaps by a beekeeper not knowing where she is, or putting a frame down carelessly or something like that. It does happen. And yeah, I think, Sarah, you were indicating on the picture there. And then also there are superseder cells, which are often, superseding is a really good way to replace your queen. That the bees do a good job of making another queen for themselves, typically, as long as they're healthy. And approximately think it's about a quarter of all colonies will replace the queen through supersedure.
The only thing I'd
And
add to
so
that is,
when you're making that intervention, oh, Bill?
the only thing I'd add to that is quite often there are two queens in a colony, and beekeeper doesn't realize that. You can find one queen, think you've made the colony queenless, but at certain times of the year, the bees are superseding, and there can be the old queen and a new virgin, or the old queen and the newer queen, and then eventually the bees will neglect the older queen and she'll die.
Okay, so then, yeah, I have two follow-up questions. One is about looking at the queen cells. Like, what does a good looking queen cell look like? Like I was showing these are along the bottom. Here's a cup. Here's an emerged queen cell. Actually, you can just see the little hatch she's come out of, and this one is still intact, or at least if it's been stung, the workers haven't cleaned it out yet. And these look good to me. You know, and this one, I mean, it's older wax, so it's not as yellow, but that's just the age of the wax. But shape-wise and size-wise, and you know, sometimes the bees will put them in funny places, and then I've kind of squished them as a beekeeper. So, like when you're looking at a supersedure cell or a swarm cell that you're deciding to keep to move in a merge to requeen another colony, could you get into that a bit?
Sure, I think that definitely a larger cell is preferable because the queen will have developed well in there. And you also, for your own sake as a beekeeper, you want to select, I typically leave 2 cells so that if one is not good, the other one, I've got a better chance with it. I have 2. But you want to make sure they're in a place that you're not going to destroy them as you replace the frame or as you do any other management or manipulation, so that you have them up. Typically, I like to either keep the 2 on the same frame, 2 good sized cells that are on the same frame, or on 2 different frames but facing each other, so that there'll be no problem with the first queen finding the other one and doing.
So if we have this circumstance where we have a really healthy hive, it's booming, and we're seeing signs that it's going to swarm, and instead of just culling all of those cells, we're going to remove them, save them, and take them to another colony that needs a queen, or to create another colony perhaps. And so our next page is a merge technique that we've used for that, which is one option for requeening that we regularly use. So can we move into that, and the other ways after that, sort of introducing a caged queen, but ways that we would replace a queen within our backyard operations?
Yeah, occasionally we'll find a colony that for one reason or another is queenless. And if we've determined that it's definitely queenless, no virgin queen, no mated queen in it, we'll merge it with a colony that does have a queen. Or it could be a nuke that you've used as a mating nuke, and it has a mated queen in it. And we do that by using a piece of newspaper. It's making a couple of small slashes in it, and that gives the bees something to chew through and to mingle. You don't want to put them together without any paper in between it, because there's a good chance they'll fight and they'll kill your newly introduced queen. So the paper is a good introduction method to combine a queenless colony with a queen right one. And then later we'll discuss some of the other ways to requeen a colony, but that one has a high degree of success as long as you have a queen-less colony and a queen-right colony.
Yeah, I love this one, and we've had some really great results with it this year. So this was actually a hive this year at the Yukon shelter and transitional housing, where we have a community apiary. And as you can see, like it looks okay maybe, but this is not a booming colony. This is right after we merged it. So when I came back to the colony with the team in the spring, we found like a very small cluster, less than 4 frames of bees, and no queen. But the colony beside it did have a queen, so we merged them using this newspaper and coming in, and on this sort of diagram, I think these are slashes, but we cover it entirely with one sheet and then make some slashes so that the bees can come and chew. And this was a fun picture just because it's of these pandemic times, and with Trudeau kind of being chewed here by the bees. But really successful. And those 2 colonies then, all of those bees got a chance to survive now that we've given them a queen. And we've been able to do that in place using that method, where if we hadn't slowed them down like that, the queenless colony might have rejected that new queen. And what is that? Like, why is that? Why would they reject a queen when they know they're queenless?
Because the introduction would be too quick, and they're not used to her pheromones, and there's a good chance that they would be aggressive towards her. So it's just better to slow down the process.
And I'm moving on to another photo here, Phil. This is again at our Italian Cultural Center yard, and this was a queen cage. So another introduction method, or can you talk a little bit about what you're doing here and what's going on?
This type of cage is not one that you'd find commercially. I make these cages out of 8 mesh hardware cloth. And the reason we use them, we carry them with us. And then if we're going through a colony and we find a queen, we'll often just put her in this cage and put the plug in the other end so that there's no chance of us killing her while we're manipulating the hive, and then release her when we're finished. The other thing we use it for, if we're catching newly mated queens, we mark them, and we put the marked queen in a cage like this, and put the plug in, leave it in there for a minute or so the paint could dry. If we were to put her directly into for instance, a JZBZ plastic cage, there's a good chance she would smear the dot on her back because they're just not big enough. So it's just a holding cage for her to dry. We don't normally introduce queens to a colony in a cage like this. It can be done, but you want a slow release, and so you want a cage that has queen candy in there. They'll give them about a 2-day release period, and you can't really do it in this type of cage.
So I have a different kind of cage in the next scene here. I also have a video of this that we can move to, so I'll share the audio so that we can hear it. But this is the more commercially available kind of cage that folks might be more used to seeing. And what are we using it for here? I think this is starting to get into some of the details.
This is it's called a battery box, and this is where we've got our newly mated laying queens that I've just taken out of one of our small mating nukes. I've checked that she's laying, and then I've marked her in the separate cage that Phil was just showing, and then I put her in to one of these plastic cages. And there you can see the blue sponge there. That's just, I put a little bit of water on there, and that's just to keep moisture in there so they don't get dried out. And then after I put the queen cages in there with the new queens, then I shake some workers, preferably young nurse bees, into the battery box so they'll take care of the queens. So then I just can close it up and off I go.
Nice. I'm gonna play this video because I think the sounds of colonies and the sounds of workers when they have a queen that they're happy with are an important thing for beekeepers to sort of understand. So I have a sound of this this very box here. So I'm gonna leave the sound on and. Diane, you didn't hear that. Did anybody else not hear it? Oh, well, that might be a problem for another time. Sorry about that. I'm not sure why. I can hear it. It's lovely. I could buzz it for you, but it's like an energized buzzing that is calm and that is consistent. And so these are like excited worker bees. I wonder why that's happening. Anybody have experience with Zoom and know why sometimes videos don't play audio? If you do, please say something in the chat and help me. But
I can just describe what we're looking at here. These are the same cages, the JZ, they're called JZBZ cages. And they're in a rack. That orange piece is a rack that the tube of the queen cage fits into. And this was placed into a queenless colony. And we refer to it as a queen bank. And we usually add a frame of emerging brood every week. We often feed it. There's no queen in the colony. And so we introduce these queens in there, and they will look after them in that colony until we need them.
I found it. So I'm going to play it again. I had to share my computer sound. So I'll just try it again so we can hear it with their nasonomic. Was that coming through for people?
Yes.
Yes, awesome. Okay, great. Because I also want to play just that other sound of the piping virgins. Would that make sense to share that as well at this time?
Mm-hmm.
So I just found a YouTube because I couldn't find this in my own archive. So this YouTube, this person here has done a great job of documenting some.
So that they can get together. Here's her neighbor.
So yeah, I just wanted to share that for a very different sound. It's quite a rare one to hear.
You can also hear that.
Maybe you want to describe that sound too.
Sarah?
Yep.
You can also hear that in a queen bank. The mated queens will make that sound also in their individual cages.
And I believe that's their wings. Is that a truth? Do I know that? Is that the wings of the queens vibrating, or how are they making that sound?
I believe so. On the comb, I think they can do it on the comb, but in the cage, I believe it's their wings.
Yeah, beautiful. And then the other sound, which is a very different one that the bees will make related to queens, is called a roar. So would you maybe describe that a bit? Like what is the sound of a roaring hive, and what does that mean when you open your hive and it's really loud?
Well, it usually means they're queenless, and it's probably because I know what it means that it's a bothersome sound, but it definitely is kind of at a pitch that puts me really on edge.
Yeah.
And it's just the sound of discontent to me. Phil, do you have any more insight on that one?
No, sometimes you can duplicate the sound if you give your colonies too much smoke and you've been in it for a long time. But if you open up a hive and you hear that soon after you open up the hive, just a buzzing sound, the bees are just making a noise to indicate that they don't have a queen.
Yeah, and it sort of grows, doesn't it? It kind of rises, and often I think that we like intuitively feel that sound is a different one, and then you spend all this time, like when I was maybe a younger beekeeper, I hear that sound, I'm like, well, I don't know, and then I keep looking through and I find no queen, and I really struggle, and I'm going through all my frames like 3 times to really like ascertain this thing that I could have known right away. And that now, like when I found that colony that I was showing earlier, this one up at the Yukon, that was quite weak, it immediately was like this, you know, this like sound of them just, yeah, showing their discontent, showing their confusion and their worry, I think. So we wanted to move into sharing some of the queen rearing that we're doing. Everything that we've been looking at and chatting about so far has really been around backyards and around merges and things inside of colonies that we might see with like 2 hives in our backyard. So Hives for Humanity, we have a few more colonies than that. We currently have 50 in the city and 20 out in Richmond. And we've been able to set up some mating yards where we have high populations of drones, and we've been able to do some work at grafting at the Italian Cultural Center. And so what we wanted to do with this workshop is, yeah, share some of the sort of general of queens and things you may have already encountered, but then also share some of this work that we're doing that is very timing specific and maybe not on a scale that you might immediately want to get into, but that there are these like elements to pull from it that I think can inform our own backyard beekeeping and our own understanding of bees. So I will start moving into those, and that's what we'll move through for the next while. So yeah, I think Leslie, do you want to start us off here, or Phil, whichever makes sense?
Yeah, so on the left side with the frame that is divided, this is a frame of drone comb that we put in quite a while before we need about 40 days ahead to make sure that we have enough drones in the area. And so that this bar is just split so that if we also wanted to use this frame as Varroa control, we could just cut out the drone brood on the bottom, but we haven't been doing that this season. We've been using it to just make sure we have a lot of drones around. And then on the right, that is our, I believe that's our cell starter, in which we basically, probably this is the cell starter before we reduced it to one box. We are, essentially, we're going to make sure we have a ton of bees in one box that, and we're going to remove the queen and have them build out the queen cells that we've grafted, the queen cups that we've grafted into. So you can see we're working with a very healthy colony, and we're going to put a lot of bees into it to have them make some great queen cells.
Yeah, and maybe I'll skip to this one because you're starting to talk about it. So do you want to continue?
Yeah, I think this is a close-up of the cell starter after it's been already put together, and that's pollen on the top, because during queen rearing, we're making sure to feed the cell starter and the cell finisher a lot of pollen and some syrup. We just want to make sure they always have access to some syrup. If they have too much syrup, they'll build wax between all the queen cells, and it just makes it more difficult to remove. And then on the right-hand side there, those are the wax cups that we graft the larva into, and we try to get, there I am on the left grafting in the graft mobile, as we refer to Bill's car. We cover it with a tarp because it's quite difficult to see them. You want to get really tiny larvae just hatched. 12 to 18 hours from hatching is optimal. So they're really small, and if we darken the car and then use that light that's in my left hand, then we can more easily see which ones we're getting. And then on the right, that's, I've just put the bar of newly grafted larvae into the frame for queen cells, and I'm going to put it into that open colony down there, which is the cell starter. You can see there are a lot of bees in there.
Yeah, it's a packed hive, especially when we contrast it to that hive that we were showing that was needing the merge, which is definitely one of the benefits of having multiple hives in any operation, like including a backyard, so that you can see that contrast. But it's really extreme seeing like how packed with bees. What a beautiful colony, and necessary, right, for very strong, so that all of these queen cells are going to be paid attention to.
Yes, exactly. Because we put 30 cells in at a time for them to draw out. And then I think another slide will show what we do with them at the next step.
This is an example of the cells. These are, it's hard to say what time this was. It's probably when we were separating them. In order to take them off to put them into nukes. Now you notice these cups are plastic cups as opposed to the ones that we saw on the previous slide, which were wax. These plastic cups you can buy, and they're couple of advantages to them and some disadvantages. One is that if you want to keep your the lines of your queens separate, let's say you graft from different queens, you can use the different colors that these cell cups come in to keep them separate. You can also see how much royal jelly is left in the cell by looking at the base. You can actually, those bases are sort of translucent and you can see the royal jelly in the bottom. And so it gives you an idea if they've been well fed or not. If there's extra royal jelly in there, you know they were well-raised cells. The disadvantage of these is that they're plastic and they're very difficult to reuse. That's why we use mainly the wax, dip the wax cups ourselves, and we dip it from wax that come from our own colonies so we don't have residues of synthetic chemicals in them.
And here on the left is the bar of 10-day-old queen cells. So approximately 13 to 14, probably 14-day-old from start to finish. More like this, we've grafted it when it's just become a larva, so that's at about 3.5, 4 days. And then we wait 10 days, so it's about 14 days. So she's gonna emerge from these cells in 1 to 2 days. And we just cut the whole bar of cells off through taking that sharp knife underneath the wax where it's attached to the bar, and then we just separate them. And then on the right, this is a picture of one of our very small mating nukes. They're, I'm not sure of the dimensions. It's probably 7 by 7 inches. Phil, I'm sure you know exactly, but this is, the green on the right-hand side is a pheromone strip of queen mandibular pheromone to keep, especially with the first round of mating nukes, to keep the bees in this tiny little mating nuke, which is not really the most hospitable box for them. So we needed to help them be convinced to stay there. You can also see at the bottom of that picture that we have a small feeder in there as well because they need some food at the outset. So we've left the gap because we're going to shake some bees into that gap between the frame with the comb.
Okay, and I'll describe how we go about this process. You can see on the right, we have these small nukes stacked up, the white boxes, we call them baby nukes because they're quite small. And what I'm doing is going into a colony, and we found the queen previously in this colony, either caged her or put her down below an excluder. And so we have young frames of brood up above, and the reason we want young frames of brood is we want to shake young nurse bees into these nukes. So I'm taking out frames of brood with nurse bees on them, and then I'll shake them into that green Tupperware container there. I think we have a video that shows that, don't we, Sarah?
Perfect, Phil. Yes, we do. I called it raisins because you were describing the bees as little raisins. So there we go.
Yeah, so here I am shaking them into the container, and Leslie is spraying them with a fine mist of water that keeps them from flying out. And young bees don't tend to fly that much anyways. It's another frame being shaken in. And then I try to get the bees all in one corner. And I use this specialized spam can here to scoop out. It's about 1.5 cups of bees. And then these are placed into the baby nuke, like so. And then Leslie was putting in a frame with a queen cell on it, the frame with the pheromone strip, and then another frame and filling the feeder halfway. And we put the covers on, and then we would load them into the truck and take them out to the mating yard.
Like so. Yeah, and one of the things that we did that you can see in our photo here is we marked them differently and painted them white at the start, and then Phil, you made these little screens, and our community members painted these flowers on top. So yeah, why did we do that? aeration and visibility for the queens? Why is that important, and what were we trying here?
Well, we want to make sure that the queen goes back to the right house because she would not be welcome at any house. So we wanted to differentiate, and as you'll see later when we're at the showing the mating yard, we faced these baby nukes all different directions so that the queens would orient accordingly and come back to the correct one and recognize their hive by the colors as well as the orientation. So this is the mating yard at the Sharing Farm Society where I also work. And this is a hazelnut orchard that we have there. And one of the main with early on in this process was they were overheating. So the first pallets we had put in partial sun, and then we gradually moved them further back into more shade because the boxes get quite hot. We had that a few really hot days, and it was right around the time when the virgins were out to go on their mating flights. And I found a massive swarm of probably, I don't know, 7 or 8 virgins and a lot of bees in it. So that was kind of a bit of a fiasco. And then we've perfected since then. We've put more ventilation into them, and we did have inner covers on all of them before, which also kept them warm. We were worried about having them be too cold. So we took those off once the weather settled more. So they have more ventilation and less insulation that's warming insulation. So
And Diane was just asking about the baby nukes and also asking about the cages earlier. So definitely has an eye for the things that Phil has custom designed for us, you know, with his experience. But these are actually gifted to us, these baby nukes, Phil. And so you know the story better than I do. But who made these?
They were made by a beekeeper in the Okanagan and then purchased by another beekeeper in the North Okanagan. And I came across them a few years ago. I bought 4 of them and had them on the balcony of my condo, and they did real well after I gave them more ventilation. The first round had absconded from those too. The virgins had left with the bees. And so it's more of this type of nuke is used more in commercial operations. They're a little finicky. They can run out of feed too quickly. You can feed them too much. If the temperature's not right, they can abscond. You have to go into them when the queens are mated to take them out. Otherwise, they get too crowded and there's no room for the next queen to mate. So I would suggest for people raising queens on their own, they just use a 4-frame nuke box or divide a standard 10-frame box into 2 and have 4 frames on either side. It's just much easier to manage to use standard equipment than to use this specialized equipment.
Yeah, and so this is the 4-frame nuke box that we were also using for, I think, well, what was that being used for? What's going on here?
that's the bigger queen in there. The white frame is the isolator frame. So we put the queen that we've selected for breeding from, we put her on a frame that has, doesn't have anything, it's all drawn out, but there's nothing else on it optimally. And it's a really good area for her to lay a lot of eggs that will be easy for us to see when we want to graft. And we put, the isolator frame goes over that frame and imprisons her on this frame. So she has to lay on that frame. And then we take her off after usually about 5 days, and she's laid up the whole thing.
And who were we mating from? Like, who was the queen who we take her to that one frame so that we know we're harvesting her eggs? How did we choose her?
This was the queen that actually headed the colony that we made into the cell starter. The cell starter is what's referred to as a queenless, free-flying starter colony. So it was a strong 2-story colony that had wintered well. We removed the queen, put her in this nuke, and then slowly jammed all the bees and the brood into one brood chamber that was queenless, and that was that became our cell starter. So she had a good brood pattern. She'd wintered well. We didn't have any other older records on her, but this was the first breeder we chose this year.
Nice. Usually like hygienic behavior and that pattern, part of that, being well mated is part of that. Can you talk a little bit more about brood pattern and what that meant when you were choosing her?
Well, we just look for a fairly solid brood pattern, not a lot of missed cells. We really haven't selected for hygienic behavior that requires some sophisticated tests.
Mm-hmm.
So we look for an absence of disease, and we also look for gentleness, because a lot of these colonies are going to be in populated areas. So we don't want bees that are going to be stinging a lot. So it's best if you can keep records over a number of years, then you can select queens whose offspring produce a good crop, a winter well, a gentle. The longer you keep records, the more progress you can make. Although it's difficult to make progress in an area like this because your virgins are mating with drones from all different colonies. If you're able to have isolation, either on an island or in a mountain valley or inseminate, you can progress much quicker in selecting and actually having a breeding program.
Just removing my cat quickly there. I do want to play a video. It's just another one that is online from a documentary that was made that shows a queen bee mating in flight. And I also just, like, I'm asking that question. I'm, you know, like, we have so many choices as beekeepers, and I think that, you know, how we choose to beekeep and the practices that we choose have an impact, like, both on our communities and the way that we share our knowledge and welcome people in through beekeeping. And the way that it can connect us to our land and our food systems, like through the sharing of honey and through, you know, starting to become fascinated with pollination and plants as the food of these bees, and, you know, the pollen and the diversity of pollen that they rely on, etc. But yeah, there's choices in how we manage the bees, and I think one of the choices really is raising our own queens and raising our own colonies. And if we aren't doing that for ourselves, you know, finding beekeepers who are doing that. So, like, what to you, Phil and Leslie, are the benefits of buying local in the beekeeping scene, you know, as well as in our groceries, or other scenes, other choices we can make with how we purchase?
I think it's, from the bee perspective, the locally raised queens and colonies are much better adapted to our particular ecosystem and have survived our cool but not cold and wet, soggy winters, things like that. And then in terms of the humans involved in agriculture, supporting those folks who are doing agriculture, I think that's really important to local food security.
Yeah, and I'd add that anytime you order a queen from a distance, that queen is going to go through a certain amount of stress during shipment. Temperature changes, and her cessation of egg laying is not all that beneficial to her. Also, the queens often come from a different hemisphere. They're on a different season than we are. It's often the end of their season when they may have fewer drones that are really necessary for mating. Anyone that exports queens to Canada has to abide by CFIA regulations, which require less than 1% varroa count in their colonies a month before they ship. And so that means they're often using synthetic chemicals to get their mite levels down so they can export, but those chemicals have an effect on the drones and on their queen rearing too. So, and also you get a reduced environmental footprint if you just either raise your own queens or buy cells or queens from a local beekeeper.
And there was another question for Diane, from Diane, sorry, about what kind of queens these are. And we aren't currently selling locally, although if all of our queens come through, we might be later in the season. I think that's right.
Yeah, we'll have some more to sell soon. We're just requeening our own colonies right now.
Yeah.
These aren't they're not they're a mix of, I'd say, Italian and Carniolan, maybe a little Caucasian. It's really difficult to get pure strains in Canada because everyone has so many different bees. So we don't raise queens according to color. We just look for the characteristics that we think are best for this area.
So I've just put it onto this slide, sharing some resources and ideas around bees and beekeeping, and also around queen rearing. Just as we're kind of coming to the end of our hour, although we will stay on for more chat and more questions. But just wanting to share this, like our Hives for Humanity website, where there's stories about our programming and images of our beekeeping, and where we share our calendar and newsletter and more opportunities to be involved, and also our socials to sort of see like day by day moments from our yards and from our bees and from our queen rearing process. Always wanting to make a plug for the plants. It really is, I think, our responsibility as beekeepers to be growing food for our bees. And so some really great resources, both locally and more further afield. But Earthwise Society in Delta and West Coast Delta, and then Xerces Society, just a wealth of information. Also always, to be thinking about the other bees and thinking about how honeybees can be an access point into understanding actually like the impact of our world, of our human communities on all kinds of bees. So on those 20,000 species, on the 463 wild species in BC. And these are really 3 really great resources for learning how to identify other bees. From Bee Friendly on the North Shore, the Environmental Youth Alliance, a really beautiful pollinator guide that they built, and the Native Bee Society, which I was mentioning Leslie is a board member of, that was just founded, that are also hosting gatherings online that recently around wild bees and habitat. A coloring sheet, if anyone's into coloring, this queen bee's mating flight, which I will play shortly. It's quite beautiful and really shows it in detail. And I'll email these out to the Eventbrite tomorrow as a follow-up. And then a real depth of videos around queen rearing that the Strathcona beekeepers have put together. Actually, I'm gonna copy them in case any of you are really keen right now. Nope, it's not letting me do it. I'll figure it out later. I will email it. So yeah, just a wealth of information there. And like I mentioned, if anybody wants to get involved at the Dunbar Club, if you came to us through that community tonight, Diane, who has been a friendly face and question asker on this gathering, she's on the other end of that email and is welcoming of new folks. So yeah, just to also say a thank you to Dunbar for the support in making this happen, and the Westside Neighborhood Small Grants and the Kitsilano Neighborhood House who put some funding towards Dunbar supporting this work, and also to take a minute to thank Leslie and Phil both for sharing your knowledge. Queen bees are just it's so complex, really, and I love being a part of this world that they're in, and I often have like dreams of queen bees. So I wish you all dreams of queen bees as you leave tonight. And yeah, I will switch and share this video, and then I'll open it up for people who want to stay on the chat. So this is just a 1 minute, and this is again online from this More Than Honey documentary that was made. And yeah, it's on this Verge website is where I found it today.
Sarah, before you push play, just that Border Free Bees has a lot of resources on their site as well.
Yeah, I can add that one in. Actually, it's a good one to add. There's such a wealth. So, yeah, thank you for sharing. And I will press play here.
Velkommen til plassen til Sikkerhetsskolen. Der så! Siste filmdronen.
mating ceremony. When the queen returns from her maiden flight, she keeps 5 million sperms in her sperm bag. In there, they can stay alive for 8 years.
Nice. So yeah, really, really great action there. Phil or Leslie, like how, when I watched this just earlier today a couple of times, like how does the queen keep flying with that drone on her and times 16 to 20 times? Like what is happening there that she can handle that?
Well, that's why you want a strong, well-raised queen. Uh, the better conditions under which they were raised, you know, and they've had good nutrition, they actually become strong flyers and they're able to mate with as many drones as they can.
Okay, well, I am going to see if I can open this up. I'm gonna unmute everyone. And so if there's any questions, you know, sort of to be spoken, now is the time and we can hang out and chat like we're around an apiary together.
And surprise, surprise. Phil and Leslie, I was wondering why you're no longer cutting out the drones. Is it just because you need them for this project or because you've decided that practice isn't working for you for some reason?
Just for this project, we want to make sure we have ample drones from good, healthy colonies.
Yeah. And that was mainly in the spring. We put these into our colonies, these drone combs in I believe late March, early April, because it takes 40 days between when a drone leg is laid and when that drone is sexually mature. So we wanted mated drones by first and second week in May, so they have to be laid in late March. And we don't really need to do this anymore. Now it can be used as a way of varroa control just by cutting these out every couple of weeks.
Yeah.
I mean, it feels like a good opportunity to ask the question of what are we doing for varroa control there? And also, Phil, how did you build this? Because this is another Phil Laflamme custom.
Well, it's not my idea. This came from Randy Oliver in California, and I think it was in one of his scientific beekeeping columns in the American Bee Journal. And this was specifically for varroa control. And the top section is just a 2-inch piece of standard wax foundation. And then the bar underneath it is actually a top bar that's turned upside down. So the groove is facing up and I cut the tabs off the end so that fits in there. And then what the bees will do is they'll make drone worker cells out of the top part because there's foundation there, and they'll fill it with honey. And the bottom section is just open. There's nothing underneath that second bar, and they'll draw a comb in there, and it'll all be drone-sized cells, and the queen will just fill it up with drones.
And you can also, sorry, go ahead.
Go ahead, Leslie.
You can also just do this simply with an empty frame with no foundation and glue a couple of popsicle sticks in the top just to give them a guide, and they'll fill it up with drone comb as well. So it's just much easier.
And we paint the top bar blue so that we know it's there. What you don't want to do is you don't want to leave these in and have them producing a lot of drones all summer. And we also usually mark it on the outside of the hive, the date we put the frame in, and also in a notebook. So you know you want to come back within a couple of weeks and cut all this out.
Right. And really, I mean, we were talking about timing earlier and the 16 days that it takes, that compressed time with all of that royal jelly for the queen to emerge as an adult. And we didn't get into the 21 days that it takes a worker or the 24 days that it takes a drone to emerge as an adult from when it was laid as an egg. But it is that 3-day window that the varroa are exploiting, that we are using to manage the varroa. So would you just get into that a little bit too, just to explain like why we would want them to build all this drone and then why we would cut it out?
We want them to build a drone comb because varroa prefer going into drone cells because it has a slightly longer development period, 24 days instead of 21. And so you get a higher reproduction rate of a varroa mite in drone cells, and they're attracted to drone cells. So by cutting it out after the drone cells are sealed and getting rid of it, it's called a biomechanical method of controlling varroa mites. Another good way to control mites is just to split your colonies and make nukes. You're interrupting the brood cycle, the development cycle of the varroa mites, and you're leaving it. Your split is broodless for a certain length of time, so there's no brood for the varroa mite to enter and reproduce.
Yeah, go ahead.
Are these frames easier on the hive or just easier on the beekeepers, so you don't have to freeze them and bring them back? Or is it too difficult for the bees to clean out all of those cells? Does this somehow make it easier for the bees or just for the beekeeper?
I think it's easy for the beekeeper, because if you use that green drone foundation, you've got to take that out, either scrape it off or put it in the freezer and put another one in. This one, all I have to do is carry around a bucket with you, cut this off underneath that bar, put it in the bucket, and if you have chickens, you can feed it to your chickens, or you can put it in compost. To me, it makes it easier for the beekeeper.
Sure. Okay.
Also useful to be able to look into, sample some of the cells and see if you have varroa in there. So it's another way you can just kind of get a sense of the mite load in a colony.
Just as an aside, I always found it a good indicator too. If your colony doesn't have a queen in it, there will be no drones laid in these. So you can just take out this. On occasion, I've just taken out this drone comb, and maybe it's been built out, but there's no eggs in it. And there's something wrong with the colony, because the queens just jump on these right away and lay into them. So if they haven't done that, you should go further into your colony and see what the problem is.
Yeah, I do think this keys into another conversation, just to circle back, and these methods of control of the varroa that are physical, and so we're making splits to give brood breaks so that there's nowhere for the varroa to be, or we're cutting out places and feeding them chickens or composting them, so that we are removing where we know the varroa really like to lay, because they get that extra 3 days of their gestation, would we call it that? I don't even know, where that foundress mite can lay her eggs and mate with her eggs, and then make more young and then take over our colony. These are the chemicals that are options out there. So we are making choices to do physical and cultural measures before we do chemical ones. And then when we make chemical choices, we're choosing organic chemicals, so the formic acid in our beekeeping. So just wanting to name that a bit and think about our choices in integrated pest management, and in controlling pests and pathogens. Leslie or Phil, I don't know if you want to add to that, if there's other choices that come to mind. If not, I might play our outro video, which is just another one of a queen. I'll pause in case anybody has a sort of final question, or Phil or Leslie, if you have any thoughts on that you want to add, any last minute gems.
Hello, I was just wondering if I could ask a question. I'm a relatively new beekeeper.
I didn't catch that. I don't know if it was breaky for anybody else.
Okay. Yeah, I do. I'm a relatively new beekeeper, and I have 2 colonies. And one of my colonies, I haven't been able to see brood for a little while. And please, no judgment. But I actually have a really hard time seeing eggs. I kind of never can see eggs. On the 11th of this month, we saw some, I think you called, there's supersedure cells that were not on the bottom, but on the frame. So we left them there hoping that they would hatch and produce a new queen. But I haven't seen anything yet. Should I buy a new queen, or is there another method, or should I wait a little bit longer? I've heard that you can take a frame of brood from our good hive and put it into the other hive. So I'm just wondering what might be the best thing for us to do.
For sure.
You might have a queen in there who hasn't mated yet, although probably there's been good enough weather in that interim. But if you have trouble seeing eggs anyway, I mean, you could do the test that we mentioned earlier, which is putting a frame of open brood, and put it into the colony you're not sure about the queen. And then if they don't have a queen, they will start drawing cells from that.
That's always the test that I use too, is putting in open brood, making sure there's like those young eggs, because no matter what, that's not going to be a bad thing if you've, we do a light shake, right? Like we don't put the full frame of adult bees in, but if you're taking bees from the center, they're mostly nurses, shaking off most of them, so you don't have it fully covered, and then putting it in to the colony you're unsure of. And then you can tell by the response the bees have to those eggs in that brood, that open brood. You can tell by their response if they have a virgin queen who isn't mated yet, they're not going to need to make emergency cells. But if that virgin queen was eaten by a bird or caught in a rainstorm or swatted by somebody or whatever it might have been, if she didn't make it back mated to the colony, those bees are going to be really enthusiastic about having that brood, because that gives them a chance again. I would say too, like we use this black foundation, and it really makes the eggs visible. So depending on what kind of foundation you're using, if you're having a hard time going to black foundation, makes it way easier to see. And then also there's an angle so that can help too, which is like sun over your shoulder and bees angled, what is this, like a 30 degree, something like that, like tipped away from you so that the sun is hitting the bottom. And then you can just be looking for the glisten of the young larvae in the royal jelly. So maybe you already know those, but those are kind of the two things, that black foundation, that I would suggest.
Okay, thanks so much.
Yeah, for sure. Okay, one more pause in case there's questions, because we could probably talk about queens for days. Okay, so I am going to play this other video then, and this will sort of be an outro. So yeah, thanking everyone again, and thanks for all the feedback and all those bees and hearts in the chat. And we have been recording this, so we will share it both at the Dunbar site and also at the Hives for Humanity one. And we will email it out to you too, so you can share it with friends if you'd like to, or re-listen to some of the descriptions from Phil and Leslie. So,
Thank you all.
thank you. Okay. Yeah, I'll play it again because I wanted to say that the reason I chose this as an outro video was just because there's that watching those bees that are around Queen and the way they respond to her and circle around her. You know, I want to be thinking about that in our communities and thinking about how we care for each other and how we offer that circle of community and of care. And I think there's so much in the bees that we can learn from, and so much that we can apply to our lives, especially in times when so many are struggling and when so many are excluded from care. So yeah, wanting to send everyone away with that thought. We will see you, I hope, around, and enjoy the beauty of your bees. Okay, good night. Thanks all.
Good night.
Good night, Phil.